Let's learn about control of respiration today. Respiratory cycle, which includes inspiration and expiration, is spontaneously generated by neurons located in the brainstem. In eupneic state, breathing occurs without conscious initiation with rhythmic discharge of the impulses. It may also initiated voluntarily by the cerebral cortex, which bypasses the respiratory centers in medulla and directly affects the respiratory motor neurons in the spinal cord. So the elements of the respiratory control are a central controller, which is pons and medulla, and the effector, which is the respiratory muscles, and the sensors, which include chemoreceptors, mechanoreceptors, metaboreceptors, and lung receptors like irritant receptors and J receptors. The chemoreceptor senses the signal and gives a feedback to the pons and medulla. The pons and medulla modifies the rate and type of breathing, and it sends input to the ventilatory pump for the respiratory muscles. The central controller can be voluntary or involuntary. The voluntary control is from the cerebral cortex, which sends signals to the corticospinal tract and to the respiratory neurons in the spinal cord. This happens during singing, speaking, and breath holding. The involuntary control is under the control of the brainstem. Normal automatic breathing is triggered and controlled by the pons and medulla, which consist of poorly defined collection of the neurons. The central controller can be divided into pontine center and medullary center. This is the midbrain, this is the pons, and this is the medulla. The pontine centers are pneumotaxic center and apneustic center, which fine-tunes respiration by regulating the timing of inspiration and expiration. Therefore, the pontine center smoothens the respiration process. The medullary centers are dorsal respiratory group of neurons and ventral respiratory group of neurons. The dorsal respiratory group of neurons plays an important role in inspiration. The upper part of the dorsal respiratory group of neurons is known as Pre-Bodzinger Complex, whereas ventral respiratory group of neurons plays role during forced expiration and sometimes during inspiration. The medullary center generates rhythm, whereas pontine center smoothens the rhythm of respiration. Now let us see about the function of the pneumotaxic and apneustic center. The pneumotaxic center is located in the nucleus parabrachialis or medialis. The function of the pneumotaxic center is to limit inspiration by inhibiting the apneustic center. Therefore, the pneumotaxic center acts as an inspiratory switch off, which results in premature switching from inspiration to expiration. The pneumotaxic center plays an important role in control of the rate and depth of the respiration. Whereas the apneustic center is located in the inferior pons. It increases the firing of the inspiratory neurons in medulla by activating the dorsal respiratory group. It delays the inspiratory cutoff switch. The pneumotaxic center inhibits the apneustic center, whereas the apneustic center activates the dorsal respiratory group. So coming to the Pre-Bodzinger complex, the Pre-Bodzinger complex are synaptic connections with the dorsal respiratory group of neurons. They act as a pacemaker of respiration. It is located in the upper end of the dorsal respiratory group of neurons. It rhythmically drives impulses to the phrenic. The underdevelopment of Pre-Bodzinger complex can lead to sudden infant death syndrome. Now let's see about the medullary centers, which includes dorsal respiratory group of neurons and ventral respiratory group of neurons. The dorsal respiratory group of neurons are located in the nucleus tractus solitarius. It plays an important role in inspiration. When the DRG gets activated, the action potential travels through the reticulospinal tract, phrenic nerve and intercostal nerve, activating the respiratory muscles. The ventral respiratory group of neurons are located in the nucleus ambiguus and retroambiguus. They are actually silent during quiet breathing, and it is activated during the forceful expiration, for example during exercise. To summarize, Pre-Bodzinger complex is a pacemaker of respiration which generates rhythm, activating the dorsal respiratory group of neurons. The activation of dorsal respiratory group of neurons results in phrenic nerve discharge, which activates diaphragm, thereby initiating the inspiration process. The apneustic center activates dorsal respiratory group of neurons, thereby maintaining the respiration. When the tidal volume reaches 500 to 750 ml, the stretch receptors are activated, resulting in the activation of the vagus nerve. Now the vagus nerve can directly stimulate the pneumotaxic center, thereby inhibiting the inspiration, because pneumotaxic center inhibits apneustic center, or the vagus nerve can directly inhibit the apneustic center, thereby limiting the respiration or inhibiting the inspiration, preventing the overinflation of the lung. Now let's learn about sensors. Sensors include central chemoreceptors, which is located in the brain, and peripheral chemoreceptors, which is located in the carotid body and aortic bodies, and the mechanoreceptors like pulmonary stretch receptors, respiratory muscle receptors, and proprioceptors located in the joint and tendon, and metaboreceptors, which are the chemoreceptors located in the skeletal muscles, and other lung receptors like irritant receptors, J receptors, and bronchial C fibers. The central chemoreceptors are located in the ventral surface of the medulla near the exit of the 9th and 10th nerve, which is glossopharyngeal and vagus nerve. They are more sensitive to H plus and increased arterial CO2. When the arterial CO2 gets increased, it diffuses into the CSF and reacts with water to form carbonic acid, which dissociates further into bicarbonate and H plus ions. This H plus ions activates the dorsal respiratory group, which sends action potential to the spinal motor neurons, thereby activating the respiratory muscles, resulting in hyperventilation, which expels the CO2. So coming to the peripheral chemoreceptors, they are located in the carotid body and the aortic body. The carotid bodies are located at the carotid bifurcation, which is supplied by the glossopharyngeal nerve of the 9th nerve. The aortic bodies are located above and below the aortic arch, which is supplied by the vagus nerve. There are two types of cells present in the peripheral chemoreceptor. They are type 1 are glomus cells, which stores dopamine and are stimulated by the hypoxia. The type 2 cells are known as supporting cells. The peripheral chemoreceptors are sensitive to hypoxia, hypercarbia, and decreased pH or increased H+. When hypoxia occurs, the oxygen-sensitive potassium channels in the glomus cells are closed, which results in increased potassium inside cells, causing depolarization. This increased potassium causes activation of the calcium channel and calcium influx, resulting in release of dopamine from the vesicles. The carotid body and the aortic body through the 9th nerve and the 10th nerve activates the dorsal respiratory group of neurons, resulting in hyperventilation, which expels out CO2. So coming to the effectors, the core inspiratory muscles are external intercostal muscles and the diaphragm. The external intercostal muscles elevate, whereas the diaphragm expands the thoracic cavity. The accessory muscles of respiration are sternocleidomastoid, which elevates the sternum, and pectorals major and minor, which pulls the rib. Whereas the core muscles of expiration are internal intercostal muscles, which pulls the rib down, and the accessory muscles are abdominal muscles and quadratus lumborum, which pulls the rib down. Thank you.